Good morning, everyone. I'll be doing a journal presentation. The topic of my presentation or journal will be new strategies for lung cancer diagnosis and treatment, applications and advances in nanotechnology. It was the article was published in Biomed Central BMC Journal 2024 by Jia Kipeng et al. From Weifang People's Hospital, China. It's a Shandong Second Medical University from Weifang. So, starting with the introduction to lung cancer as per the journal, uh, we all know the reported incidence of 2.2 million cases of lung cancer per year with the 1.8 million deaths, still the number one in the global cancer data. And of two types, non-small cell lung cancer and small cell lung cancer, which accounted 85% and 15% respectively. And the diagnosis methods, we have for screening, we have low-dose CT scan. For screening, we have low-dose CT scan. But the exact recommendation for the screening purpose is not yet established. And we have another modality, that is pathological modality, by acquiring the tissues in the diagnosis. The low-dose CT scan is supposed to have high false positive up to 86.4%. And furthermore, on lung cancer, there is a high EGFR mutation rate in East Asian lung adenocarcinoma, that is 38 to 50%, according to the article. And osimertinib plus chemotherapy, they have 83% response, but only 57% of 2-year disease-free survival. And even the immune checkpoint inhibitors like PD-1, PD-L1, and CTLA-4, they show limited progression-free survival gain as per the Checkmate 722 trial. So there is the definitive gap in the early detection, definitive gap in the exact treatment strategies, which has a lot of room for improvement. And this is where the nanotechnology comes in the recent times. So another thing the articles has given us is developments beyond surgery for the treatment of cancer. We also discussed about the development of carcinogenesis some days back in the topic of carcinogenesis, that in 1915, a cancer was established induced in another living organism in Japan. And since then, there is a lot of development in oncology, chemotherapy, and immunotherapy section. So we first had the chemotherapy, which targets the normal cells as well as the cancer cells both. And slowly we then had the immunotherapy, which targets to the certain immune section of the cancer cells. And now the immunotherapy is also past the recent advances. Now there is a time of the nanotechnology. So this is for the reference that from the fingertip, the video has zoomed in 10 to the power 9 times inside. The 10 small particles is in the cells. This is the protein RNA particle which is found in the cells, which can act as the carrier. So this is the nanoparticles. There are different type of nanomaterials which are. So I'm talking about the nanoparticles inside the cell. The nanomaterials can be made in the lab as well. So this is for reference. Nanoparticles are 10 nanometers in size. If we have to write it mathematically, it is 10 to the power minus 9 in measurement. So earlier the concept was the nanoparticles will go into the cancer cell and will cast some different type of light, right? Like infrared rays or photodynamic lights, and the nanoparticles will kill the cancer cell. So these are the early concept. But now the nanotechnology have been evolved, and there have been development of the nano carriers, which carries the drug and targets the specific cancer cell. And we'll discuss more about these nano carriers. So on further exploring the topic, they have also given the role of nanotechnology, that is nanoparticles measuring 1 to 100 nanometers of size, high modifiability and targeting. And this can be applied in diagnosis, drug delivery, imaging, and gene therapy. There are various types of nanocarriers. This we have to know. The lipid-based nanocarriers, solid and lipid nanoparticles, liposomes, nanoparticles, and encapsulated liposomal nanoparticles. They are different. They functions differently. Some functions as a drug conjugate. Some functions as a antibody drug conjugates. So we'll explore each according to the article. And the definitive advantage of these nanocarriers are enhanced drug delivery. That is, they improve the transport of hydrophobic drugs, they increase the targetability and bioavailability, and reduce the embolism risk. And there is also a unique nanoscale properties, means it is a small molecule which will go into the target and change into the bigger high surface to volume ratio molecule. It has a self-assembly property, physiochemical stable and controllable morphology, which makes it suitable for the drug delivery. And then tumor-specific cytotoxicity, that is surface modified nanoparticles enable targeted drug delivery, which minimizes the systemic toxicity. There will be no accumulation of the toxic drugs if we deliver it via the nanoparticles. And improved bioavailability of the drugs is also other added advantage. Sustained drug release responsive to tumor environment, as we can change the pH of the drug according to the tumor microenvironment. We can change the redox state, enzymes, and stimuli responsive systems, which allow the precise drug activation. There is a optimized pharmacokinetics, which improve the absorption and half-life of the drugs, and already been discussed in a previous presentation about the CRISPR-Cas9 technique, which is the new technique for the gene editing. In case there is some epigenetic aberration in the tumor formation, so this CRISPR-Cas9 technique is also a new nano carrier that is being used commonly. So, firstly, the lipid-based nanoparticles. These are the cationic lipids which may have cholesterol or may be pegylated, that is, polyethylene glycol may be there. It delivers both hydrophilic and hydrophobic drugs. The chemotherapeutic drugs are primarily they used to be hydrophobic drugs, which we had a hard time delivering these drugs, but these nano carriers have made it easier. And there's an added advantage of high stability, drug loading, and biocompatibility with the lipid-based nanoparticle. The example is the lipid-based nanoparticle spray for siRNA, which targeted the lung cancers is being evaluated. Another is protein-based nanoparticles, that is mainly albumin, which is commonly used even in our setting. We don't know, we may know the NAB paclitaxel, that is nanoparticle albumin bound paclitaxel, which has the higher targetable and delivery of the paclitaxel to the cancer. So advantage is of drug delivery flexibility, which makes the systemic toxicity lesser than expected. And there are also antibody drug conjugates targeting specific lung cancer antigen, but the role has not been proven yet. Another is the viral nanoparticles, that is, they can be conjugated adenovirus. They can be virus-like particles. They have high transduction efficacy. They are not limited by any barriers in the blood in the body. But the limitation is they can be picked up by the macrophages. They have high immunogenicity. The pegylated interferons that we use in interferon alpha, pegylated interferon alpha in hepatitis C, and in other cases, these are the also newer example of viral nanoparticles. Another is apoferritin nanoparticles. These are iron depleted ferritin nanogases, which induces the apoptosis if it goes to the tumor cells. The advantage is low toxicity and tumor targeting. And another, the commonly used is in lung cancer. The proven benefit has been shown from China is the polymeric nanoparticles, which is the nanospheres or nanocapsules. We now have the nanocapsule encapsulated with the osimertinib, that is biomimetic nanoparticles loaded with EGFR TKI osimertinib, also known as CMNP at OC. This is how the nano drug is written, featuring a polymeric nanoparticle core and coated with the tumor cell membranes, which goes to the tumor cell and utilize antigens on the cell membrane and targets drug to achieve the dual targeting. That is, it targets the tumor via the osimertinib, it targets the tumor via the immunogenic property. So there is a dual targeting of the tumors. Another is the metal nanoparticles. It is being used in some GI tumors like pancreas tumor. It can be gold-based, silver-based, iron-based, or platinum-based. It also helps in the imaging. Most commonly, the gold-based imaging technique is used in for the prophylactical MRI of the BRCA positive females, and limitations. Those females who are not willing to undergo the mastectomy, BRCA positive females. And limitations are nephrotoxicity or hemolysis. Example is the supermagnetic iron oxide NAP. This is also known as spion or supermagnetic iron oxide nanoparticles. Or spion CCPM means the cross-linked polymeric micelles. These are smaller than the nanoparticle, which modulate the tumor immunity and goes into the tumor selectively. So this is the list of some nano formulations that has been approved since the year 1995. The first one that was developed is pegylated doxorubicin, live dox or doxil also written. It was approved for HIV-associated Kaposi sarcoma and later for the metastatic breast cancer as well as the ovarian cancer in 1995. Then the daunorubicin was approved, and then cytosine or citarabine was approved, liposomal bound citarabine. And then the most commonly used, that is nab-paclitaxel or Abraxane, the drug. It was approved in 2005. And then the vincristine or pegylated vincristine. And now the Abraxane is now nab-paclitaxel plus gemcitabine coated Abraxane is available for metastatic pancreatic cancer. It was approved in 2017. And then the irinotecan, pegylated irinotecan for metastatic, as second line drug for metastatic pancreatic cancer was also approved in 2017. So these are the FDA approved nano drugs till now. So if we see the current status of nanoparticles based therapy in lung cancer specifically, the main challenge here is to achieve the tumor specific targeting while minimizing the healthy tissue damage. This key challenge still remains critical. We have various articles for the lung. The Spiegel et al. They did a research on squamous non-small cell lung cancer with the nab-paclitaxel for plus carboplatin for induction and maintenance. The primary endpoint was not met, but the trend was shown to be improved in overall survival. And then Sai et al. They also compared polymeric micellar paclitaxel (PM-PAC) versus solvent-based commonly used paclitaxel, and there was the improved PFS and response rate shown in 3 years research. And Weiss et al. They did a research in a stage 4 non-small cell lung cancer in a post-platinum therapy. Where the nab-paclitaxel showed promising improvement in the overall survival, especially in the geriatric patient. And other articles like Sosinki et al. Stage 3B4 non-small cell carcinoma, which shows the higher overall response rate versus SV-paclitaxel, that is the solvent, commonly used solvent paclitaxel. So these all articles, they show that these nanoparticles combined with the various other chemotherapy or immunotherapy even has a better overall survival, and some of them has also shown the better progression-free survival as well. Even though there is according to the review by In and Nieva, even though there is a marked improvement over the conventional therapy by these nanocarriers, there is still a limitations. There is no set guideline for how for the doses, and no set guidelines for about the systemic toxicity. So it requires the further innovation and optimization for these newer carriers. So now the diagnostic application, we have the nanotech enabled imaging enhancement, like gadolinium-based imaging, which improve the tumor radius sensitivity and MR contrast. Also, the perfluoro15 ground pyrethroid PFC. This is the inhalational nano carrier, which is enhanced in MRI via microphase targeting. This has been used in um, certain lung cancers as well. And in the case of another is the PEG SPO nanoparticle, that is EGFR targeted nanoparticle, which is enhanced in MRI plus ultrasound. And on the ultra small rigid platform, which is non-invasive, which shows the sub-mm tumors by the use of the nanoparticles, and fluorescence. It is used for solid pulmonary nodules as well as even the sub-millimeter pulmonary nodules before the development of the cancer. So the recent trend is towards the development of the theragnostic application, that is therapeutic as well as the diagnostic in the same setting. There have been the development of two platforms, UC ILA platform, that is up-conversion nanoparticle plus IR dye, which is the simultaneous imaging and immune therapy towards the cancer, and another the manganese porphyrin at ferrous silicate composite. There is a dual mode MRI, and the nanoparticle carrier triggered the pH based drug release in the tumor, and which acts on the tumor selectively. So there is the two newer theragnostic application of the nanoparticles. And in case of the surgical integration of these upcoming topic, that it can help in real-time biosensing for the surgical decision making. That even the Chinese, they have been using graphene oxide and gold nanoparticle-based biosensor, which detect the tumor markers or circulating tumor DNA intraoperatively after the dissection of the tumor, which guides after the excision of the tumor, which guides on further decision of the extent of tumor dissection or lymph node dissection, which helps in reducing the need of invasive diagnostics during the surgery, or the may ultimately provide for the better outcome after the surgery as well. And if we see the nanotechnology, they have been developed more in the field of oncology, that is radiotherapy or chemotherapy or immune therapy. And exploring more on that, there are some challenges in the using nanotechnology in imaging therapy. First is the hypoxemic tumor environment, and next is the cancer stem cell, which resists the radiation via continuous self-renewal. So the radiosensitizers like gold, gadolinium, or binanoparticles, they can be used to concentrate the radiation dose in a tumors, sparing the normal tissues. This has been FDA approved, and this has been being used rapidly in China as well. There is SPONC platform, which releases the iron under the radiation and trigger the apoptosis. So there is a dual act on the tumor by radiation as well as by the iron. Another is nanotech in chemotherapy. The challenges are there is the high toxicity to normal tissue by chemotherapy, and the drug resistance due to tumor heterogeneity, a change in microenvironment of the tumor, or mutation of the p53. So there may be poor drug stability and non-specific distribution of the drug according to the characters of the drug as well. And the nano solution, that is the solution by nano carriers, are the polyethylene glycol acid composite chemotherapy can be used, which goes in the tumor microenvironment selectively, or even the human serum albumin conjugated with platinum can be used to deliver the chemotherapy in the selective cancer cells. And this is the recent, that is T7 carboxymethyl chitosan based chemotherapy, which targets the lung cells with the platinum based chemotherapy selectively. And there have been some advances in overcoming the drug resistance as well. So the fluorinated nanoparticles are used to degrade the mutant p53 and induce the ER stress. And there are other carbon nanotubes and cationic lipid polymers which boost the chemistry sensitivity. And the carbon nanotube, which is bound with doxorubicin, they helps to reside the doxorubicin longer in the cancer cell, ultimately killing the resistant cells. Also, some of the carriers can act as a synergistic radiochemotherapy action, like X-ray responsive peptide nanogels. The drugs reaches to the cancer cell, but it gets triggered only during the radiotherapy, which enhances the efficacy and reduces the systemic toxicity. So another is the targeted therapy applications. Some of them have been discussed already about the uh, osimertinib encapsulated nanoparticles. The limitations of the conventional targeted therapy is sometimes there can be off-target effects despite tumor specificity. And there can be development of drug resistance. We have seen the osimertinib resistant tumor cells as well. And there can be a suboptimal tumor drug accumulation and systemic toxicity as well. So there is a solution for these as nanocarriers like hyaluronic acid, erlotinib and bevacizumab. Uh, they have the co-delivery of erlotinib or bevacizumab has been made possible for gastric tumor as well. And the bevacizumab coated ziftomenib nanoparticles, ziftomenib loaded nanoparticle with bevacizumab has enhanced accumulation in non-small cell lung cancer. Also, the red blood cell membrane coated ziftomenib nanoparticles have the proper targeting and real-time tumor image. It helps in real-time tumor imaging as well. So it also, as with the chemotherapy, the nanotechnology also helps in overcoming the drug resistance of these immunotherapy as well, such as the copper sulfide nanoparticles. This switch the bypass signaling acquired by the tumors to resist the immune system. And the EGFR TKI plus siRNA nanoparticles, these are the dendrimers, which are the dendrimers. To understand the dendrimer, it is for the smaller particles than the nanoparticles. And so dendrimers has the co-delivery with the lysosomal skip, which sensitize the resistant cells. Also, the RAS triggered release of osimertinib or selumetinib has been made possible to overcome the drug resistance of the osimertinib in case of the lung cancer. And there have been some integration in immunotherapy as well as so immunosuppressive tumor microenvironment, poor drug penetration, limited immune activation, and low response rate are some of the challenges in our traditional immunotherapy. And the nanotech enabled the doxorubicin bound nanoparticles or the PDL1 that is podophytotoxin loaded lipids which suppress the PDL1 expression and the lipid nanoparticles which carries the RNA encoding interleukin 12 which has a strong immune activation. These are some of the nanocarriers which enables the immune modulation and kills the cancer cell. So this is taken from the reference of the same article, the DNA nano robot. This is the future of the nanotechnology, which function as the cancer therapeutics in response to the molecular target in vivo. It was published by Suping Li et al. And the research was done in China. The nanoparticles, the thrombin-coated nanoparticles was delivered to the cancer cell. The nanoparticles are smaller than the tip of the DNA, and they are sent towards the tumor cell. And the thrombin-bound nanoparticles, it clots the vessel supplying the cancer, which ultimately kills the cancer. It was a test done in the mice, guinea pigs. We can say the thrombin-bound drug was injected via the tail of the rat. And before treatment, this is an illustrative diagram shown by the author. Before treatment, there is a marked shrinkage of the tumor size after the treatment due to the necrosis by a thrombin clog. Even though the future has already been the present in many of the countries, there are still the limitations of the nanoparticles. There is a reproducibility issues, which is there is a difficulty in consistent manufacturing of the uniform nanoparticles as these are very small in size, and there is a scalability challenge, which is a complicated large scale production quality control. And the toxicological and safety concern is still there, and limited targeting specificity, which is being changed by the nanobots, as we have seen. there is a reduced bioavailability at the tumor site due to the very small size of the particles, and there is a chance of interaction with biological barrier, leading to the complex interaction with immune system and biological membranes. To sum, to sum my whole presentation, these are the nanoparticle types, which have certain advantages and disadvantages being used in the lung cancer and other GI cancers. So, to summarize in 3 words, I'd say that nanotechnology, it enhances the holistic approach of the lung cancer care as well as other cancer, from the diagnosis to the treatment. So the key strength is of the specific targeting, reduced toxicity, improved diagnosis. The future is in the personalized nanomedicine. Now, there is a upcoming topic of the theragnostic, that is therapeutics as well as diagnostic in the same setting as already been discussed. And there is also a possible integration of artificial intelligence in target specific in the nanoparticles. The evolution of nanobots is an example of these programmable nanoparticles, and even the CRISPR-Cas9 technology, the one I discussed some days back in carcinogenesis topic, which helps in the flipping the mutated gene and then adding the remaining DNA particles, that is CRISPR-Cas9 technique, has been the development in AI integration in CRISPR-Cas9 technique has been evaluated in certain parts. So now the discussion points for the future could be how can these nanotech overcome the current lung cancer diagnostic gaps? We still don't have the screening tools for the lung cancer. So nanotech can be used, or it can overcome these gaps in future. Or the regulatory hurdles can be cleared in clinical use in the future, or they can be integrated with AI and precision oncology. The main problem with these new advancement is this is very easy to use in bench, whereas it is very difficult to bring these innovations in the bed to the patient side. So this is one point for the future innovation. Thank you.
Thank you, Dr. Sandeep. Indeed, it's a very eye-opening topic. I think many of us are not aware of the things that are happening in the world. So the very important thing is that he mentioned, highlighted, is the technology is being changed, where still it was the previous era where the general chemotherapy is being used, radiation therapy is being used. So somehow if we can reduce the toxicity of the systemic chemotherapy or the radiation therapy, that would be great. So from that point of view, the nanotechnology has come. So far Nepal is concerned, we do prescribe the nab-paclitaxel. That's available in Nepal. I think at higher cost, but it's available, and we have been using this. I don't know what's the efficacy. So there are some points which we should discuss. The first point, he said that yes, there is the scope for the nanotechnology. I also believe there should be the scope of the nanotechnology in the future, very near future, I guess. So are there some research articles which just compared, there is the nanotechnology or nab-paclitaxel versus general paclitaxel for lung cancer? Is there head-to-head comparison?
Yes, sir.
Some article, and what's the result?
There have been 2 articles which compares the nab-paclitaxel with solvent-based paclitaxel that were used conventionally, and they have shown the progression-free survival to be better in one article, and in another article they have shown that there is no change in progression-free survival, whereas overall survival is improved, especially in geriatric patients.
Okay, so this is the one. I think there is some hope in the future. The second thing he mentioned that you mentioned that there is a role during surgery.
Yes.
So can you elaborate how it is done? How it will help during surgery? I'm performing the surgery. There are the nanorobots. How they are going to help me during the surgery?
The concept that the authors have proposed in this article is we give the nanoparticles before the surgery, and we dissect the tumor along with the lymph node, systematic lymph node dissection. And if the nanoparticles show there is still the circulating tumor DNA, means there is a tumor cells in some parts. So we may have to do the extensive lymph node dissection, or maybe extensive extirpation of the lungs or maybe it is already a disseminated disease that we don't know of. So this is the decision making. It helps in decision making during the surgery. That is the concept they have provided.
Is there any concept that will show you that I have not done the complete surgery? I mean, you are talking about circulating DNA, it means it's already systemic disease, so we are not going to control it by just doing the regional resection of the tumor. So if it's circulating inside the blood, it means it may say that it may point to us there is a disseminated disease, though it has not been proven so far. Even if you, what's the if value of the detection of the circulating tumor DNA? Even if you get it positive, but it has not been implemented in the clinical practice. There is no such data that will suggest, yes, there are some data which shows there should be some researchers that will say that if the circulating tumor DNA is recorded preoperatively, then there is no meaning in doing the surgery, resecting the tumor. Solely the circulating tumor DNA is present in the blood. It means the patient is going to have poor survival, there will be metastasis, but it has not been taken into the clinical account so far. Maybe in the near future, it's logical, of course. Circulating tumor DNA inside the blood means there shouldn't be any point in doing the local resection of the tumor. So I think near future that will show. But my point is, you've said that nanoparticles will help during surgery. So let's suppose we are assuming tumor is localized and our intention is to cure the tumor by local excision of the tumor. So is there some way that during the excision of the tumor that will show that you are leaving behind some of the, you are doing the R plus section? Is there some way that will direct you, help you, say, yes, it's the R plus section, please go a little bit ahead. I mean, more research is required.
So yeah, so there are lipid-based nanoparticles which is shown in fluorophores, which emits light, sir. So they go and bind to the tumor cells, and they may emit light in even in the near infrared. So if we do the R plus resection, then these nanoparticles may emit light.
That's very good thing, I think. Yeah. I think that's the future. In near future, perhaps the nanotechnology will come in a good way. Key comments, anything that you'd like to say? Because if we are new to us, I think it will take maybe years to come in about. Let's hope it will come in. That's a good thing. Nanotechnology case help, okay, input case, suggestions, or anything.
What about nano-based imaging? Is like better than the conventional imaging we have, like PET scan or other imaging?
There has been no research on comparison it with the conventional PET scan or MRI, but there was one article which showed the gold-based MRI for the BRCA positive females who are not willing to undergo mastectomy to detect the cancer before the happening of the cancer.
One more thing, in the literature, did you go through some points like if the patient has the degenerative disease of the spine? So they put some nanoparticle containing some scaffold or some cells, primitive cells that were regenerated into the neurogenic tissue, etc., and that will help in removing the paralysis of the patient, right? So is there something like that, so that is going to change the genetic material or genetic mutation that has caused the tumor inside the body of the patient? Is there some references or some research in that?
Research has shown the use of CRISPR-Cas9 techniques, such as a gene editing tool. Suppose we have a mismatch repair gene, MMR gene, which is not working, and the cancer is seen in the colon or somewhere else. So we can use the CRISPR-Cas9 technique to clip those DNA which have the MMR gene, mutated MMR gene. So remaining DNA will be MMR free. So if you cure the cancer, the resulting tumors from the MMR defect will be.
That's a very good point. Why I raise this question is because let's suppose somebody is having genetic hereditary polyposis cancer. Okay?
Yes, sir.
So he has right now developed the colonic cancer. It means in the near future he might develop some GI cancer or pancreatic cancer, etc. So if you are doing the repair of the MMR gene, is free from the defect by the nanotechnology, so there will less chance that you will develop in this future some other tumors at other sites. So I think this is a very good point. Genetic change, genes can be modulated by the nano. So very good. Okay, Sadiva.
future will be of like nano-based technology with enhanced with AI.
No surgeons, AI.
So,
China has made some hospitals containing 50 AI robots.